Hello, and welcome to a new edition of H.E.L Explains with your host, Dr. Mario Torres Rodrigo. And today we're going to be talking about a fascinating topic. Today we're going to delve into the gut microbiome. But the first thing that we need to ask is, what is a microbiome? Well, a microbiome is the total amount of microbes that inhabit an ecosystem. And we can regard our own bodies as an ecosystem themselves. If we think about the human body is made of tens of trillions of cells. That is a huge number. We are talking about a 1 followed by 13 zeros. And what is the microbial makeup of this amount of cells? Well, actually, if we think about the amount of cells that inhabit our body compared to human cells, we are talking about a 1-to-1 ratio. So we have approximately the same amount of human cells than microbial cells. However, one thing that we need to bear in mind is that microbial cells are much smaller than human cells. So actually, despite the fact that they make this huge amount of numbers, they are only up half a kilo of the total body mass of humans. However, this number can vary, obviously, depending on the mass of the person and so on and so forth. So the microbiome is, as we said before, the total community inhabiting an ecosystem. And when we talk about microbial community, we are not only talking about bacteria. There are other members of this microbial community, such as archaea, or fungi, such as yeasts, protozoa, viruses. And characterizing the community is not a mean feat. Actually, it's quite a resource and time-consuming task that has evolved over time. 20 years ago, for example, that sequencing wasn't as developed as it is nowadays. Characterizing the community was quite a tedious task that involved cultivation. So you had your sample, you had to grow your microbes, and then you would do a number of things to try to characterize these microbes, such as, for example, trying to identify what kind of metabolisms they had, or if you had the means to extract the DNA and then do some very basic sequencing of certain sequences in the DNA of the microbe that will tell you what kind of microbial species or what kind of microbial taxa you had in your hands. However, in the last few years, the techniques have been evolving, and the next step was using 16S RNA. So that is a phylogenetic marker that cells have. A phylogenetic marker is basically like an index in the cell that you can look and say, so this sequence is very familiar to this other species. So this is the species that it belongs to, or maybe if it's a new species, or this is the species that is most similar to. And then more recently, the technique has evolved to the point that we can just extract the whole DNA in a sample, and then we can just do what we call shotgun metagenomics, in which we just break all the DNA into short sequences. And what we can do afterwards is just rebuilding these small sequences of the whole of the genome, so we can identify all the different communities that are inhabiting this ecosystem. The research of the human microbiome has become a priority, to the point that in 2007, the Human Microbiome Project was created, aiming to promote research characterizing bacterial, archaeal, and fungal communities associated with humans. Some of the results from this network has deemed that actually the gut microbiome is particularly important. And on the next few minutes, we are going to spend some more time talking about gut microbiome. But just as a highlight, it has become so relevant that InfoGest was created. InfoGest is another network that aims to harmonize methodology used in research linked to the digestive system. So let's just jump and talk a bit more about the gut microbiome. The gut microbiome is a dynamic ecosystem. What does this mean? Well, any ecosystem existing that contains organisms, especially microbial organisms, are going to be massively influenced by physical-chemical parameters that are going to shape the microbial communities. One of these physical-chemical parameters, especially in the terms of digestive system, is going to be the diet. Whatever we're going to be ingesting is going to affect our microbial community. But also we're going to see differences along the gastrointestinal tract. Just to give you a couple of examples. The small intestine, it contains some oxygen because it's further up, so it's closer to the mouth, so some of the oxygen is going to come with the food. And also it has a quite fast transit, so the muscles around the small intestine are going to be pushing the food that we have ingested down. The result of this presence of oxygen plus this fast transit is going to result in a fast growing facultative anaerobic community. These are a lot of big words, but just to break it down, this means that we're going to have microorganisms that can use oxygen to respire. They're going to be growing very quickly, and in case that there is no oxygen, they're able to use different means to survive. However, if we move further down into the colon, so just the last part of the gut, we're going to find is that there is no oxygen because the microbes living upwards have been just consuming this oxygen as they were digesting the food. So there's no oxygen. And also the muscles in this part of the body are going to be slower. So this means that the community is going to be different. In this case, we are not going to have facultative anaerobes. Instead, it's going to be completely anaerobic community. So a community that doesn't use oxygen and that potentially oxygen is pernicious to them. So how do these microbes survive if there is no oxygen? Well, they have another range of pathways they can use. So for example, they can ferment, or they can use different substances instead of oxygen to respire, such as sulfate. So obviously this microbial community inhabiting the guts and the human, that is the host, are going to have a relationship. What kind of interactions do they have? So one of them is that the microbial community inhabiting the gut can protect their human host. How do they do this? They are able to outcompete against pathogens because obviously they are residing in the colon. They've been there for a very long time. They are very much used to the conditions in the gut. And they're going to be the ideal conditions for them to grow, which means that they can grow faster than the potential pathogens that are going to attack. But they also have other range of things that they can use, for example, producing antibiotics. A second type of relationship between the gut microbiome and the host is that microbes have metabolic capabilities that humans can only dream of. Because they are so small and so simple, actually they are very specialized and they have pathways that we don't have. And they can help us produce certain products that our human bodies can't. For example, vitamins or factors such as anti-cancer factor, anti-inflammatory factors, these microbes can produce that we cannot. But also, there can also be negative interactions. For example, if we find that the microbial community inhabiting the gut are deprived from nutrients, they need to find their own ways to survive. And this means that in some cases, what they can do is affect the host. So for example, the mucous barrier that surrounds the colon is very rich in certain sugars that normally they're not available for microbes. But if the microbes inhabiting the gut microbiome feel themselves that they don't have enough food, they're going to start attacking this gut barrier and they're going to degrade it. And this means that some negative effects can happen. So, I love my 3 takeaway messages from the video. So I guess that the first one is that humans are never alone. We have a huge microbial community inhabiting our bodies next to us. Secondly, gut microbiome is a very important topic, and as a result, there are a number of organisms that have been created to promote gut microbiome research. And third, the relationship between the gut microbiome and the host is very complex and it can have positive or negative effects. And that's it from me. My name is Dr. Mario Torres Rodrigo, and if you have any questions, if you have any queries, please don't doubt and reach for us. Thank you very much.